Hello, my name is Dr. Charles Knight. I am professor of Bio 414 Evolution, and this is a series of lectures that I decided to make, which I'm calling 10 Lectures on Evolution. That's my series. I'm from Cal Poly, from the Bailey College of Science and Mathematics in the Department of Biological Sciences. So this is my first lecture in this 10 lecture series, and I hope to see you 9 more times. This paper is titled "The Reconstruction of the Human Amylase Locus Reveals Ancient Duplication Seeding Modern Day Variation." The paper was carried out by a number of people split between 2 institutions, and I'm going to talk about the authors just a little bit, just to give you a sense of where this work was done and who these people are. Faiza Yilmaz is the lead author, and Omer Gokman is the last author. Those are 2 important positions of authors on the paper. And then there's 2 other authors who, if you pay attention to the superscripts, contributed equally. So these first 3 authors are also equal participants and should be considered the primary author or the first author. In addition, Charles Lee is an important funder for this work. Him and Omer were the PIs at 2 different institutions that I'll talk about on the next slide. So I could say the work was led by Faiza Yilmaz and Kwando Kim in the Jackson lab, and Professor Gokman and Chari Kella from the University at Buffalo in New York, and others. If you don't know, the Jackson Laboratory in Connecticut is, well, they say, leading the search for tomorrow's cures. It's a very important biomedical institute, kind of like the Howard Hughes Institute of Medicine, and they're really doing a lot of really cutting-edge work that relates to evolutionary biology. And I think what's cool about it is that institute touches on evolution a lot, because finding the cures for diseases involves a comparative approach from other animals, not only in humans. And also, of course, it's easier to do research in non-human subjects, and so they're doing a lot of research on mice and even plants at the Jackson Laboratory, even though their goal is to search for the cure for human diseases. So I'm going to read, I've got 3 take-home points from the paper. First, the fact that there's variation in copy number in the human population. Just any gene, copy number variation. If you don't know, there's variation within our population. Some people have more copies than others, other people in our population. We're not all exactly the same. That shouldn't be too surprising, but for me, this is really one of the main takeaways from the paper. Second, that paleogenomics, sequencing the genomes of extinct or humans that lived 20,000 years ago or even older, is possible these days, and that can illuminate the timing and processes of molecular evolution, especially in gene families like the one that we'll talk about today. Third, Neanderthals used fire to cook starch, but they only had 3 copies of the amylase gene, which we'll see is the enzyme that breaks down starch, and they only had 3 copies, whereas humans, modern day humans can have up to 30 copies, and so that's a big difference. And you wonder whether that might have contributed to Homo sapiens outcompeting Neanderthals about 40,000 years ago. You wonder, and I think actually in the paper that phrase or that sentence was cut from the paper, I think because the editors probably thought that was too correlational. It didn't make it into the paper, but that's my assumption, that's just my conjecture, is that perhaps that could have contributed. Now I'm going to read the abstract. I think the abstract's very important. Scientists have thought long and hard about the words that go into the abstract, so I think it behooves us to start with the abstract. So I'm going to read it slowly here and try not to make many comments about the abstract, and I'll follow up in a minute. Previous studies suggested that the copy number of the human salivary amylase gene, AMY1, correlates with starch-rich diets. However, evolutionary analyses are hampered by the absence of accurate, sequence-resolved haplotype variation maps. We identified 30 structurally distinct haplotypes at nucleotide resolution among 98 present-day humans, revealing that the coding sequences of AMY1 copies are evolving under negative selection. Genomic analyses of these haplotypes in archaic hominins and ancient human genomes suggest that a common 3-copy haplotype dating back as far as 800,000 years has seeded rapidly evolving rearrangements through recurrent non-allelic homologous recombination. Additionally, haplotypes with more than 3 AMY1 copies have significantly increased in frequency among European farmers over the past 4,000 years, potentially as an adaptive response to increased starch digestion. Now I'd like to go over some of the important vocab from the abstract. I'm circling 4 important phrases or words or acronyms: AMY1, haplotype, negative selection, archaic hominins, and non-allelic homologous recombination. Now I'm going to define them on what I call my glossary notes slide. We'll start with AMY1. AMY1 is the salivary amylase gene. It breaks down starch, and it's going to come up over and over again in this presentation. Haplotype. It's a multi-locus genotype where it's basically where the genetic basis of a trait resides. That's a haplotype, but you can have variation in a population for the haplotypes. Not every individual has the same haplotype. They may have a different combination of alleles. Negative selection is what we sometimes refer to as purifying selection. It's selection for the same type, or it purifies the population. I don't like that word, purifying selection. That's why it's gone the way of the dodo. Instead, we call it negative selection, but in the textbooks and the first time you learned about it, you might have called it purifying selection. Hominins are individuals or individuals that lived a long time ago that are on the evolutionary branch leading to modern humans. That's what a hominin is, and so the archaic hominins are ancient humans or subspecies. Homo sapiens living 20,000 years ago, for example, would be an ancient archaic hominin. And Homo neanderthalensis is also an archaic hominin because Neanderthals contributed a lot to the Homo sapiens genome. Homo floresiensis, maybe archaic hominins, we're not sure, we're not able to sequence the genome, but that's a different subject. 3-copy haplotype. That's the 3 copies of AMY1 or amylase in archaic hominin genomes, for example. That's a 3-copy haplotype that we'll see in the genetic maps and reconstructions. And perhaps the most difficult term is non-allelic homologous recombination, which is the exchange of genetic information between 2 similar or identical strands of a double-stranded DNA molecule. Took a deep breath because don't want you to get lost in the weeds. It's not so important that you understand this specific part and how it happens, but if you do know a little bit about genetics, this is where recombination takes place during prophase 1 of meiosis 1, when the arms of a chromosome intermingle with the arm of another paired chromosome, and genetic information can be exchanged. That's one way to get this non-allelic homologous recombination, but there are other ways too that we'll see later. So I wanted to give you some backstory about the important kind of constituents that lead up to this paper. First of all, glucose is a simple polysaccharide that I'm sure you're well familiar with. Humans can easily digest it, perhaps too quickly, and it's too good. These are some molecular models of alpha-D-glucose. It's cyclical. There's 5 carbons, or actually 6 carbons, C6H12O6, and this is the ring structure. You don't need to know too much organic chemistry, but I'm just reminding you that this is the good stuff that we can easily digest. That's in comparison to starch and cellulose. Starch we can digest. It's more complex than glucose, but simpler than cellulose. You can digest starch, but not cellulose. So I put these emojis on that starch is yummy, cellulose not so much. Can't digest it. So amylase is the enzyme that breaks down starch, and amylase breaks alpha-1,4 glycosidic linkages at random points in the starch molecule. So I've highlighted with a red line these joinings between individual glucose monomers, and that's where amylase would just kind of randomly break these larger starch molecules. And they break it at the alpha-1,4 glycosidic linkage. Just to get into the weeds here, this is the alpha-1,4 glycosidic linkage between adjacent glucose molecules, and you can count up the carbons and impress yourself with your organic chemistry knowledge. Not that important for this paper, but just wanted to show it. Eventually, that leaves us with glucose, which is yummy for humans. And I want to point out that the production of glucose from starch by amylase makes rice taste somewhat sweet. Maybe one of the reasons you like to have rice instead of potatoes is that when you're digesting it in your mouth, the salivary amylase gene is already breaking down the starch in rice and producing glucose in your mouth. I'm not talking about sushi rice, where they actually add a little bit of sugar there sometimes. But just regular rice tastes somewhat sweet if you think about it. It's not like a candy bar, but it's a little bit sweet in your mouth. Try to make that observation next time you consume rice or just regular rice. I've got kind of 4 points to make continuing on that don't have good graphical illustrations. Humans have 2 versions of amylase. Amylase 1 or AMY1, which is expressed in the salivary glands, and AMY2, which is expressed in the pancreas. Starch-consuming animals have undergone independent bursts of AMY gene duplication from the ancestral AMY2-like gene. So the pancreatic amylase gene was the first one, and there's been duplication and diversification into amylase gene that's expressed in the salivary glands. And so the rest of my course in Bio 414, we talk a lot about developmental biology and how a gene could go from being expressed in the pancreas to being expressed in the salivary glands, and it must have to do with Hox proteins giving it the genetic coordinates or the geographic coordinates in the body to be expressed in the salivary glands. Certainly, those Hox genes would act as transcription factors that would bind to regulatory sequences in the copy AMY1 that are unique to the AMY1 copy that target it for expression in the salivary glands. That's a little bit tangential to this presentation, but it's part of the Bio 414 course that I teach. A great ape specific duplication resulted in the salivary amylase gene, AMY1. So there was a duplication in the great apes. Other animals don't have the salivary amylase gene, but great apes had this duplication, and now there is one that's expressed in the saliva, which opened up new food sources to great apes. And now, in Homo sapiens, we have 2 to 30 copies of AMY1 per diploid cell in a human. So that's a pretty amazing or expansive or large expansion of this gene family in Homo sapiens. These are some data from a study that was done at Cornell University, where they just went to students on campus and said, hey, can I have a saliva sample? And then they sequenced the genome or this locus around AMY1 and were able to measure the number of copies of each student had. So this is a frequency distribution, and on the x-axis is the number of amylase copies, and this is just for AMY1, and CN stands for copy number. So on the x-axis it goes from 2 all the way up to 30. You can see individuals that have more than 12 are fairly rare, but more than 2 is very common. Now, this is reference 8 from the target paper that I'm focusing on today, and it kind of led me into a aside, and it almost derailed this whole presentation because reference 8 was really intriguing, and they did something that was, I just can't help but tell you, it's amazing, but it really sidetracked me. The title of this reference paper is Human Salivary Amylase Gene Copy Number Impacts Oral and Gut Microbiomes. And if there's something I know about college students these days, as soon as you say the word microbiome, everybody gets excited. So I thought I'd try to look into this a little bit more, and I did, and I found some really gross stuff. They took the stool samples from some of these students. Well, they had volunteers in the high amylase copy number. On the right-hand side, you can see there's a number, this individual has 8 copies of AMY1, and there's people in the low category that had only 2 copies. And then they took stool samples from students in those categories and then did a fecal transplant into mice. And this is where I kind of lost track of time. I didn't have enough time to go into what the results of that were and why exactly they did that, but that's amazing that they did that. But I'm just going to leave that there, and maybe students will follow up on this in another companion lecture series, which are the student presentations in this course, and we'll learn more about this study perhaps. But it got me a little sidetracked, and I needed to go back to the source. So I want to go back to our source. This is the graphical abstract that was produced with the target paper that I introduced at the beginning of this presentation. Some papers in Science and Nature and PNAS and other journals have what's called a graphical abstract sometimes, and sometimes they're useful. It's kind of a synopsis of some of the figures that are in the paper and basically a graphical story of the entire paper. When it's useful, I'm going to use these graphical abstract slides or figures as much as I can because it's kind of similar to me giving this presentation to a broader audience. You're not sitting down to read the whole paper. You want to get the main points. And so this kind of graphical abstract may be hopefully very useful for this presentation. Just checking my time here. It's still going. Here, I'm going to focus on the top figure first and then go down to the bottom figures. When I have this text in a box, this is kind of the take-home point of this particular graphic. What we're seeing here are 4 different human haplotypes at the AMY locus. AMY is the acronym for amylase, and this, I should say, is, well, it's at the AMY locus, and they may be AMY1 or AMY2, with structural variations such as duplications and inversions. That word structural variation comes up a lot in genomics, and this is an image of 4 different haplotypes. The haplotype at the top has 3 copies, all being transcribed in the same direction. The second haplotype down, H2A2B2, is a haplotype that has 6 copies. They are all being transcribed in the same direction, and really it seems like this cassette of 3 has been duplicated. So now you have 2 cassettes of 3, 6 total. Haplotype H3 has 3 copies going in one direction with one of the alleles going in the reverse direction and then another copy of that pink allele. That's a different structural variation, a different haplotype. H5, haplotype 5, has a total of 8 amylase copies, and you can see the structural variation there. They're going in both directions, and it's it's different than the other haplotypes. And that's part of the big finding from this paper. This figure is a simplification of one of the main figures from the paper. This is figure 1 from the main paper, the target paper, but frankly it's too complex to include in this presentation, I felt. The summarization that we just saw is much more digestible, I think. So when we can, we'll use the graphical abstract. Now we'll look at this other piece of the graphical abstract, and I'm going to make it bigger so you can see what it is. And the statement that goes along with this part of the figure is that negative selection was observed on all amylase gene coding sequences. And frankly, this is just a colorful drawing. It's not helpful at all to conclude that this statement that they make, that negative selection was observed on all amylase gene coding sequences, when actually they have a figure in the paper that does a pretty good job of this. So we're going to jump to that figure. It's more difficult to understand. Well, it's I should say it's easier to understand than this, which you can't get anything from, but it introduces a concept that I'm going to have to go in depth to all the way back to the textbook so that you can understand what this thing is called DNDS. But this figure is not helpful. This figure is, and now though I have to explain it in piece by piece. I'm going to... All right, so on the previous figure there was a metric called DNDS, and I need to explain what DNDS is. It sounds like calculus, but it's not. It's a ratio of non-synonymous to synonymous mutations. It's a molecular test for selection. I'm pointing you to the location in our textbook, page 265, that explains what DNDS is and what we can learn from it. Basically, the ratio, if it's less than 1, that's when replacements are deleterious. There's fewer non-synonymous mutations than you would expect, and that is negative selection or purifying selection. When DNDS is equal to 1, evolution is neutral, and when DNDS is greater than 1, then that's when replacements are greater than synonymous mutations, and that's when those replacements are advantageous. So when we go back to figure 2a, we will be looking at first the single nucleotide variants and the amino acid replacements along each branch in the amylase gene family phylogeny. And then we'll look at DNDS ratios. Stick with me. So I want to help you along here with some glossary notes that I think are going to come up. Paralogs are gene copies within a species. For example, copies of AMY1 within humans are paralog or paralogous, and it's within these paralogous or orthologous genes that we can measure DNDS, because you have to have some comparison to know if the mutation led to an amino acid change or not. An orthologous gene is a similar kind of comparison, except you're comparing the genes between species. That's a different kind of comparison that happens a lot in comparative genomics. DNDS, again, is the ratio of non-synonymous to synonymous single nucleotide mutations in paralogous or orthologous genes. Big words, I know, but there's no way around it. Those are words that are used a lot in comparative genetics and genomics. Negative selection, again, is when DNDS is less than 1, and aka purifying selection. Non-synonymous mutations are deleterious, and then positive selection is when DNDS is greater than 1, and that's diversifying selection, and non-synonymous mutations are favored in that case. I want to look at one figure from our textbook that illustrates DNDS, and it involves the BRCA1 gene that you've probably heard of, and it shows DNDS measurements along branches in the primate phylogeny. You see humans at the top, and then chimpanzees, gorillas, orangutans, all the way down to rats and mice. But the point is that along the branch leading to Homo sapiens, you see DNDS is 3.1. That's much greater than 1. That implies that non-synonymous mutations are favored. There's a lot of non-synonymous mutations versus synonymous mutations. If you go further back, you see branches leading to branch points having much lower, 0.04, 0.5, 0.7, much lower DNDS values, implying that there's pretty much neutral evolution for BRCA1 until that branch leading to humans and chimpanzees, and in both humans and chimpanzees there's been significant positive selection for the BRCA1 gene. BRCA1 is a tumor suppressor gene, so it's not surprising that it's been under strong selection. That's why when there's a mutation in BRCA1, that's why it can lead to cancers like breast cancer. So that's just kind of an aside. Now we're going to look at the DNDS for amylase genes. first, we look at the single nucleotide variants and the amino acid substitutions along each branch. We're looking at 3 different amylase genes. AMY1 is on the bottom, it's gold. AMY2 is blue, 2A is blue, and 2B is red. Those are 2 different ancestral amylase genes from which AMY1 was derived, and you can see AMY1 is more closely related to AMY2A, but the point here is that there's a lot more single nucleotide variants in AMY1 and a lot more amino acid substitutions comparatively, although there have been more single nucleotide variants. Those numbers by themselves are not that useful. What we'd rather see is the DNDS value, and that's what we see here on this other part of figure 2A, which is the DNDS estimate for AMY2B, 2A, and AMY1. For 2B, the estimate is below 0.25, and the p-value is 0.003, so they're pretty sure about this. Way less than 1. 1 is way over on the right-hand side. It's a dotted line over there. I wonder if you can see my spot here. Much lower than 1. And the AMY2 is still less than 0.5. AMY1 still a lot less than 0.5. Overall, what we see is negative selection in the amylase locus, and that's one of the take-home points, or for their paper, big take-home point is this negative selection at the amylase locus. The next part of the graphical abstract is this structural variation, and this structural variation is, I think the graphical abstract figure is pretty good. They identified 2 mechanisms for this structural variation. Non-allelic homologous recombination, which is essentially unequal crossing over, but then we also have microhomology mediated break-induced replication. Ugh, big words, long, but what we can see is a lot of shuffling around of allele copies, copying, inversions, duplications, a lot of activity at this locus producing lots of variation. Last figure is fairly straightforward. We see that on the branch leading to the great apes, first we see the branch up to the great apes, and there's only 1 AMY copy. See, all the way at the bottom, all the ancestral was just to have 1 AMY1, and I think what they meant, well, they said AMY1 here. I remember AMY2 is ancestral to AMY1, but at least before the branch between chimpanzees and Homo sapien, they already had 1 AMY1. Chimpanzees, I would assume, has 1 copy of AMY1. And then we see, before the split between Neanderthals and Homo sapiens, there were 3 copies. Neanderthals have 3 copies. Homo sapiens have many copies. You can see on the top left that Homo sapiens has many copies with lots of structural variation, directions and copy number. You also see where agriculture came to be, and we'll see this in better detail in their next figure, which is figure 5. So this is a useful timeline, and what we see is diversification of AMY1 copy number and when it happened. It happened after the use of fire, so humans were already using fire. It was starting to break down starch with the use of fire, but it was after that use of fire in the paleontological record that we see an amplification and duplication of the AMY1 locus. You can see at T2, that's when the first initial duplication was, and that was around 800,000 years ago. Now this is complicated because this is going all the way back to Australopithecus. This is before the genus Homo evolves. If we date this divergence to 800,000 years, that would have been in Australopithecus. It wasn't until about 700,000 years when Neanderthals split off from Australopithecus. So this is ancient history. The human Neanderthal split, they're showing it around 800,000 years. I've heard more recent estimates, but the initial AMY1 duplication was before that. And we see more recent things going on where there's a pre-agricultural expansion of the gene family, and then post-agricultural expansion, more recent times, and currently at T5, or more recently, or what they're calling recent, is there's lots of variable sweeps in the population where these structural variants are being fixed, or not fixed, because there's a lot of variation. So populations vary for their copy number of amylase and their structural variation and what haplotypes they have. I hope you're with me with all those words. And we're looking at AMY1 copies per diploid cell here through time along this sequence, and you see the gradual increase in AMY1 copy number leading up until the recent times. And we also see an increase in starch consumption in more recent times. And this is coming from paleontology. So this is a nice collaboration between paleontologists, genomicists, and molecular biologists who are just really interested in how amylase works. So the major burst in AMY1 copy number increase came after a gene inversion around T3 from that original timeline, and it's unique to ancient humans but not Neanderthals. So Neanderthals don't have it. Ancient humans did have it, but the branch leading to modern humans preserved that and then went wild with it and diversified a lot more. I'm going to go back to my take-home points. The reconstruction of the human amylase locus reveals ancient duplication seeding modern-day variation. First take-home point is that simply there is variation for gene copy number in the human population. Secondly, paleogenomics can illuminate the timing and processes of molecular evolution in gene families. And third, that Neanderthals used fire to cook starch, but they only had 3 amylase gene copies compared to our up to 30 copies, which may have contributed to the dominance of Homo sapiens 40,000 years ago when Neanderthals went extinct. Thank you for listening, and I hope you're back for lecture 2, 3, 4, 5, all the way through 10, and maybe check out the student series as well. Thank you.